In this video, I am going to break down digestion and help you absorb absorption from a nutrition perspective and in a way that makes complex science easy to swallow. The process of digestion begins with mastication, also known as chewing. This mechanical action stimulates saliva release from the salivary glands in the mouth to moisten and soften food so that it can be crushed with up to 90 kilograms of bite force into a manageable mush. While about 99% of saliva contains water, it also includes electrolytes, mucus, and immune cells. Carbohydrate lovers will be pleased to know that an enzyme, salivary amylase, kicks off the chemical phase of digestion in the mouth by targeting starches in foods like rice, potatoes, peas, and beans. We also can't forget about lingual lipase that targets short and medium chain fatty acids attached to triglycerides that are found in butterfat, coconut oil, and palm kernel oil. Poor old protein doesn't have any chemical digestion assistance in the mouth. We have to do all the mechanical work by chewing our food to pieces. No matter what food we eat, the actions of the mouth serve to increase the surface area available for the major chemical digestion that's awaiting just beyond the pharynx. To get there, the tongue must push our food mush, also known as a bolus, towards the back of the mouth to initiate the involuntary action of swallowing. This bolus movement results in the lifting of the larynx and subsequent covering of the trachea by the epiglottis to literally prevent us from inhaling our food. Nobody wants aspiration pneumonia. Once the bolus slides past the epiglottis, it is headed down the esophagus via a wave of muscular contractions known as peristalsis that literally squeezes the partially digested food mash until it reaches the muscular gatekeeper of the stomach, the lower esophageal sphincter. As it relaxes, the bolus is allowed passage onto the acidic environment of the stomach. This sphincter can commonly malfunction by not fully closing and results in a sensation known as heartburn. That's just the stomach's way of saying to the cells of the esophagus that acid rules and the stomach is strong. Speaking of the stomach's strength, it has 3 layers of muscles arranged in a longitudinal, circular, and oblique fashion that enables the stomach to be a real powerhouse when it comes to smooth muscle contractions. These peristaltic contractions continue the propulsion of the food bolus, but also serve to expose more surface area of the bolus by crushing it into smaller bits so that the chemicals have better access to all of the bonds holding the macronutrients together. It's the job of the gastric glands of the stomach to release hydrochloric acid to continue the chemical phase of digestion. The downside is that the salivary amylase and lingual lipase, initially released in the mouth, are now deactivated because proteins of any origin, whether they're from food or released by our body, become denatured or unfolded from their original shape, which causes them to lose biological function. They are down and out for the count. But digestion isn't because the muscular contractions by the stomach continue to mix and mash our bolus with the freshly squeezed hydrochloric acid. This helps to continue breaking apart carbohydrates into maltose and dextrin, which are small polysaccharides. These same powerful contractions also help partially emulsify fat so that they can play nicely with water and not end up like your oil and vinegar salad dressing. Scrumptious for a few minutes, but then they go their separate ways. Interestingly, there's another enzyme called gastric lipase, which is released by the chief cells of the fundus of the stomach that continues the challenge of breaking apart the short and medium chain fatty acids found in butterfat, coconut oil, and palm kernel oil into diglycerides and free fatty acids. Pepsin, also released from the gastric glands, breaks apart longer strands of protein called peptides into smaller sizes. One last point of interest on our journey through the stomach is that mucus is released to protect the cells of the stomach from its own inhospitable environment. Coincidentally, being hostile is also the stomach's way of keeping party crashers like bacteria from spoiling your digestion festivities. Oh no! The celebration continues as the food mash, now known as chyme, passes through the pyloric sphincter into the initial segment of the small intestine known as the duodenum. Chyme fragments as large as 3 millimeters or about a teaspoon can pass through at a rate of about 2 times per minute. The presence of acidic chyme in the duodenum results in the release of a hormone called secretin, which is secreted by the S cells of the duodenum. There sure are a lot of S words in digestion, right? Can you think of any that I've missed? Secretin is important because it signals the pancreas to secrete bicarbonate into the duodenum to help neutralize any acids carrying over from the stomach to help prevent cellular damage and allow the continuation of chemical digestion by enzymes. Speaking of which, secretin additionally stimulates the pancreas to secrete a variety of enzymes designed to specifically target each of the macronutrients. Notable pancreatic enzyme mentions include alpha amylase that targets starches, pancreatic lipase and colipase that break apart triglycerides into monoglycerides, glycerol, and free fatty acids, phospholipase that chips away at phospholipids, cholesterol esterase which chases after those cholesterol esters, and finally zymogens, which are dormant enzymes that eventually become activated to karate chop those proteins. Even after all that work of secreting bicarbonate and enzymes into the duodenum, the pancreas continues with the addition of electrolytes and water to aid in all that chemical digestion. But wait! We're not done yet! Secretin has a buddy called cholecystokinin that is secreted by the eye cells of the duodenum when fats and proteins are present. Cholecystokinin also puts the pancreas to work to secrete enzymes specific to fat and protein digestion that we mentioned earlier. It's all about doubling down. Why not have a backup plan to make sure that the correct message is being delivered to the pancreas? Food is present and we need to extract nutrients. But cholecystokinin isn't just part of a redundant system, it also stimulates the gallbladder to secrete bile into the duodenum to help emulsify fats so that they play nicely in the watery environment of the intestines. The bile salts contained within bile help break apart lipids further and forms a wrap around long-chain fatty acids, monoglycerides, cholesterol, phospholipids, and fat-soluble vitamins. This wrap is known as a micelle, which is basically a taxi that allows easy transport of hydrophobic lipids through water. Geez, the duodenum is such a busy place! If you thought we were done, think again. The duodenum also has a series of enzymes located in the microvilli. Those are the hair-like projections extending from the intestinal cells known as enterocytes. We have disaccharidases that target carbohydrates to release monosaccharides, monoglyceride lipase that targets lipids to release fatty acids and glycerol, and a variety of peptidases that target oligopeptides, tripeptides, and dipeptides to release amino acids. Now that's a system that ensures we can extract as many nutrients as possible from chyme. By the time chyme passes through the ileocecal sphincter to the large intestine, only fiber, insignificant amounts of fat, and about 1% of protein consumed constitutes what is now referred to as feces or stool. That's how efficient the human gastrointestinal tract is at extracting nutrients from the food we ingest under normal physiological conditions. Absolutely extraordinary. But wait! Feces don't just exit the rectum and go for a swim in the toilet. What? They provide fermentation material for the resident bacteria or microflora that inhabit the large intestine. These are good bacteria that make things like lactate and short-chain fatty acids that can be used by enterocytes for energy and even circulated back to the liver for ATP production. In addition, phenolic compounds are produced that can act as antioxidants against oxidative stress. A symbiotic symphony that also includes the production of hydrogen, carbon dioxide, and methane gases that are sure to temporarily make the air in your surroundings a bit more odoriferous. Now that's what I call a gas. But Dr. Santo, you've only described digestion. How do we absorb all of those nutrients that we just spent about 10% of our daily energy expenditure releasing? Yes, indeed, we still need to discuss how those nutrients are internalized so we can help maintain whole body homeostasis. Let's dive in and begin in the mouth. Basically, no nutrients from food are absorbed in the mouth. Insignificant amounts of ethanol can be absorbed directly into the bloodstream via sublingual and buccal capillaries, and of course some medications. In the stomach, absorption is once again limited to trivial amounts of ethanol and medications, but can also participate in the minor absorption of water. There's not a lot of absorptive action going on until we hit the small intestine. It's like the Las Vegas Strip of the gastrointestinal tract. That's where the majority of the absorptive action happens. As nutrients are swimming in the lumen of the small intestine, they are pulled in by the sweeping action of the microvilli across the brush border membrane of the enterocytes. From these intestinal cells, nutrients are transported across their basolateral membrane, allowing them to enter the hepatic portal circulation, which diverts all nutrients to the liver. Monosaccharides cross the basolateral membrane using both passive and active mechanisms, but easily dissolve in blood because they are water soluble. They can travel the great highway of the hepatic portal vein until they reach the liver, which decides the fate of these carbohydrates. Lipids are a bit more complex. Glycerol and short and medium chain fatty acids cross passively across the basolateral membrane and use a protein taxi called albumin to travel through the hepatic portal vein until they are offloaded at the liver for distribution. Long-chain fatty acids, monoglycerides, cholesterol, phospholipids, and fat-soluble vitamins, which were wrapped up as micelles, are repackaged in the chylomicrons and exit the enterocyte via the lacteals of the lymphatic system because they are just too big to cross the basolateral membrane. The thoracic duct of the lymphatic system carries these chylomicrons to the large veins of the neck, allowing them to enter circulation for distribution throughout the body. Amino acids, dipeptides, and tripeptides use facilitated and active diffusion mechanisms to cross the brush border membrane, as well as the very intricate and complex series of amino acid and peptide-specific transport systems to gain entry into the enterocyte. When it comes to protein, the enterocyte is like a very exclusive restaurant. You have to be the big cheese to get in. Within the enterocyte, peptides are further broken apart into amino acids, which can be used for the energy needs of the intestinal cells, transamination, and protein synthesis. To cross the basolateral membrane, similar transport mechanisms are needed in order for amino acids to enter the hepatic portal circulation. Since amino acids are water-soluble, they easily swim through the bloodstream until they reach the liver, where the liver decides what to do with them. But what about water? How does that get absorbed? Now that we have our juices flowing, water is an integral part of digestion and absorption. About 50 to 60% of the ingested fluid load is predominantly absorbed in the duodenum and jejunum segments of the small intestine. The large intestine also participates in the absorption of water, but it only accounts for about 15% of the total amount of ingested fluid load. With that said, the large intestine can absorb as much as 80 to 90% of the fluid entering the colon. That's phenomenal regulatory control, enabling us to avoid wasting a precious nutrient. The mechanism for absorption is mostly passive, depending upon the solute concentration and subsequent osmotic pressure, and typically happens in the presence of a high to low concentration gradient. Kind of like what happens when you add salt to cucumbers. Water follows the sodium. Wow! The human gastrointestinal tract is intense to study, right? How can I keep all that juicy information straight? My suggestion would be to eat your favorite food, and as you are doing so, think about what it takes to digest the macronutrients into smaller bits, followed by how those released nutrients can make their way into the bloodstream. If you have any questions, please reach out to your instructor. We are all here to help. Have a healthy and productive day.